When we last left off, I talked about the fact that when we break down food through cellular respiration, when we release energy, we do it in small steps so we don't explode as we do it. We have to control that release of energy. If all the energy were released at once, it would be catastrophic. And one way we do that is with a molecule known as NAD+. NAD+ is an electron shuttle. So what happens is NAD plus 2 electrons, and I told you hydrogen usually comes along for the ride with electrons. So it becomes NADH. So NAD plus is reduced. Remember, if you gain electrons, you're reduced. Is reduced to NADH, and then NADH can go somewhere else and then release those electrons again, so they release more energy. So one way we do this is by grabbing electrons and not letting them complete the reaction immediately. We grab them and then we release them in another reaction and control that reaction. So NAD plus, like I said, is like a school bus. So here's NADH, so it has its hydrogen and its 2 electrons attached, and so now we can see, that's really annoying. Okay, so now we can see they're ready for transport. So from food to the school bus, and then the school bus, NADH, let me just make sure you're clear on that. This is NADH. Takes them to something known as the electron transport chain. When those electrons are dumped off in the electron transport chain, they are used to make ATP. And eventually those electrons get stuck to oxygen gas, forming water. Now, we could get rid of all these steps, and electrons could go directly from food to oxygen gas, and this would be fire, detonation, flames, right? But instead, we break it up over several steps. We release energy a little at a time, and that's the energy we use to make ATP. And since it's controlled, we don't explode in the process. This is that molecule. NAD plus seen here. Notice once again, it's like a nucleotide. We have adenosine here. We have that ribose sugar. We have 2 phosphate groups in this case, and then another adenosine stuck here. And then there's this molecule, nicotinamides stuck here. We're not going to memorize this formula or anything in this case, but what happens is electrons from your food get stuck to this molecule here. You can see that happening here. Electrons from food as well as some hydrogens are stuck to it, so when it becomes reduced, you can see another hydrogen get stuck in this area. You can see that happening right here. And then later on, that hydrogen and those electrons can be released again. So what we're able to do is release electrons in several stages, and every time they transfer it to one of these stages, a little bit of energy is lost. So up here is high energy, down here is low energy. At the very end of all of this is oxygen waiting for us, and we generate water at the end of all of that. And what's going to happen is NADH is going to dump electrons into that electron transport chain, which acts like a stair step. It harvests the energy from the electrons, and that energy is used to make ATP. So glucose is oxidized to CO2. We've already mentioned that. If those electrons were given directly to oxygen, that would be combustion. But instead, we separate these reactions out into several steps and slow down that release. Intermediate electron carriers are used, releasing a little bit of energy at a time, like going downstairs, and that prevents detonation. Your book has a very similar idea. If we mix hydrogen gas, for example, and oxygen together, explosive release of heat and energy, and you have water formed at the end. However, we can do the exact same thing, but we can put an electron transport chain in between the mixing here, so and harvest a little bit of energy at a time. We produce a bunch of ATP, we control the reaction, and we still get water produced at the end. It's just that we harness the process and we slow it down and we take advantage of it rather than just let it detonate instantly. We'll talk more about how that works later. Okay, so aerobic respiration, which is the theme of this chapter, aerobic means with oxygen around, is broken down into a few steps. Glycolysis is the first step. Glycolysis, that means lysis means to break, and glyco means sugar, so this is the breaking of sugar. Glycolysis. Then the Krebs cycle, also known as the citric acid cycle. And then the electron transport chain. For every molecule of glucose that we talk about in this chapter, we can produce about, that's what this tittle means, about 30 ATP molecules. Okay, that's important. It could be 32, it could be 28, 29, 28. We'll just say about 30. All right, now there are a couple ways to make ATP. So first of all, our building blocks are typically ADP and a phosphate molecule, which they'll usually draw like that, like an inorganic phosphate. And we have to get them to combine somehow. One way to do that is substrate level phosphorylation, which is shown below. Substrate level phosphorylation, we've used the word substrate before. That's when an enzyme is involved. That's when an enzyme grabs a hold of one molecule that already has a phosphate on it and causes that phosphate to break off and stick to ADP, making ATP. This happens in glycolysis. It also happens a little bit in that citric acid cycle, okay? But you only get a couple of ATPs this way during cellular respiration. Now, the other kind of phosphorylation in this chapter is oxidative phosphorylation. That's using oxygen and the electron transport chain, which we'll look at later, and that's where we get most of the ATP that we generate. So we can use an enzyme that just breaks off a phosphate group and sticks it to ADP, making ATP. Or we can use this process of oxidative phosphorylation, which we'll describe in greater detail later. Okay, so in the upcoming chapter, or in the upcoming lectures, I want you to pay attention to this color code. Things that are kind of this blue or teal color are glycolysis. Things that are this reddish salmon color are the citric acid cycle and something known as pyruvate oxidation. And purple items are the electron transport chain. And so we're going to break cellular respiration down into these three major categories and then look at each one in turn. And what I want you to be able to do, and there's some really good guided reading questions on this that you should use to study. I want you to focus on the inputs and the outputs and the major things that happen in those reactions. We're not going to memorize every step. Like your book, I know, let me grab your book. Your book does a pretty good job of this, but your book does have a few images that might scare the heck out of you, like the one on the bottom of this page here. We're not going to memorize every step of these processes, okay? That's for organic chemistry and courses like that. That's not what we do here. We're going to look at the inputs, the outputs, and some of the primary ideas involved, like redox reactions. So we're going to start with glycolysis in the next lecture and move our way through cellular respiration. It's going to be imperative that you take careful notes, that you read the book, that you ask yourself questions, and you figure out what's going on here. This is not impossible to understand. It just takes some work. All right, I'll see you in the next lecture.